Buenos días a todos. Muchas gracias por asistir a esta tercera plática que se engloba dentro del proyecto de la investigación científica como lenguaje universal. Quiero agradecer a la Facultad de Medicina por todo el apoyo que he recibido, así como al programa de internalización en casa 2020, modalidad virtual de la Facultad de Medicina. El día de hoy tenemos la plática acerca de nutrición y ritmos circadianos, que va a estar dirigida por el doctor Bertrand. Pido precisamente a la doctora Estefanía Ochoa que lo presente.
Buenos días. A continuación daré una semblanza del doctor Bertrand. Él es investigador principal en la Universidad Mixta 1280, Fisiología de Adaptaciones Nutricionales. En la Unidad Mixta 1280, Fisiología de Adaptaciones Nutricionales en Nantes, Francia. Es ingeniero agrónomo, realizó un entrenamiento en virología en el Instituto Pasteur para después obtener el doctorado en Ciencias Farmacéuticas en la Universidad de Tours. Realizó una estancia postdoctoral en la Universidad de Uthecht. Su línea de investigación involucra el estudio de la alteración del perfil epigénetico de la leche materna con microRNAs, el cual puede abrir la puerta a la suplementación nutricional materna para mejorar el cuidado de los infantes prematuros. Ha participado en la creación de la primera biocolección francesa dedicada a la colección de muestras no invasivas en bebés prematuros. Como especialista en tecnología tisular y celular, ha participado en la creación de cultivos intestinales 3D y 10 líneas celulares porcinas inmortales disponibles en la colección europea de cultivos celulares acreditados. Muchas gracias.
Muchas gracias.
Entonces, puede comenzar, doctor.
Buenos días a todos. So, I'm going to do my presentation in English because my Spanish is not good. So, welcome. So, I would like to introduce you to diet and circadian rhythm and an application of it, a possible path to cure or prevent diseases of civilization that I shall present: diabetes, obesity, and cardiovascular disease. An avenue to prevent such diseases is to manipulate the chrononutrition of the patient. And in the next slide, I need Sandra. So yes, thanks. I'm going to give you some definition about circadian clocks. So circadian clocks is a group of genes which are organizing our daily life, and the synchronizer is an environmental signal. Periodic environmental signal which can be understood by the circadian clock system. So the next slide, I can show you a small map of the system. So synchronizer, the best example of synchronizer is the light-dark cycle, and of course, it's the best explored and the most important in human life, because we know many things about the influence of the alternance of day and night on our physiology, because we know the sensory transducer. So in the second column from right, sensory transducer are a special group of retina cells called the melanopsin cells, and they are equipped to detect by melanopsin the transition from light to night and day to night on the reverse. And so this sensory transducer is sending the signal to the central clock of the organism called suprachiasmatic nuclei. Suprachiasmatic nuclei are located at the bottom of the hypothalamus, and they are two small masses of approximately 10,000 neurons, and they are usually presented as the central clock of the body, and they are sending signal to the pineal gland, which is producing the melatonin hormone, and melatonin hormone is the hormone telling all our somatic cells whether we are in the dark or a light phase. And of course, this central clock is entraining the peripheral clock, mainly, for instance, the liver. If we are looking the other environmental synchronizer, the second type is food feeding. And the problem with feeding is that it's not as simple as day and night cycle, because the sensory transducer of feeding is unknown. And in addition, when you are eating a meal, it's more complex as the transition between day and night. You have the frequency of the meal and the composition of the meal, but anyway, it has been shown that feeding is interfering with the day-night cycle. And if the survival of the individual is at stakes, sometimes the body can ignore the signal of day-night cycle and focus on the feeding signal to synchronize the activity of the physiology. Next slide, please. The next. Sorry. Okay. So this is to show you how the circadian clock is organized. So you have a cell, any kind of somatic cells, and inside you have a system of positive and negative element. And this system is giving in the body, in the cell, a way to obtain a clock, because you have a temporal pace created by the interaction between positive and negative element, and the system is recycled by degradation through phosphorylation. And of course, you have many external factors which can influence the functioning of the circadian clocks, but it has to be a periodic signal with a range of time relatively accurate between 22 and 28 hours. And in this system, you have the microRNA. I'm a specialist of microRNA, which are for sometimes now progressively introduced in the regulation of this system, so that you have a balance between the proteins, the messenger RNA, and this microRNA able to detect any temporal signal difference in your environment, so that you are accurately organized to distinguish day and night, but also the period of the day. And of course, any cell is sending output signal, and this output signal have consequences on the general physiology, not only on the wake-sleep cycle, but also on your humor and on the functioning of some organ like liver. Next slide. So there is three main physiological functions which within the realm of the circadian rhythm, thermoregulation. So over the day, mammals have an internal temperature which is relatively constant, but you can detect oscillation over 3%. So that means that the stability of the macromolecular complex in our body are acutely regulated by this thermoregulation. The second is the regulation of osmotic pressure. It's more important with plants. And the third is the regulation of the stability of the chemical composition. So to keep it simple, for instance, your blood, the level of your blood sugar, and of course, when you are going to the laboratory for a blood examination, the physician is advising you strongly to go early without food. And this is because we are trying to make measurement avoiding the circadian activity of the body across the day and the night. Next slide, please. So the food and the rate in the term of feeding is a complex system to study for scientists. The first parameter is the number of food intake, and in human, that means meal, but with rodents in laboratory, you can define time access of food. And of course, you have the composition of the food, and the composition can change the feeling of the organism. And of course, if you have a different composition with different influence on your physiology, you can be driven to prefer some kind of food over some other. Next slide, please. So I summarize on this slide the main pathway for food entrainment. You can entrain people by changing the daylight cycle. And in general, you need 1 hour to be of exposure to a different lighting condition to be able to entrain your body on a new cycle. And with food, it's different. And the composition of food is very important. And the best demonstration has been made on the relationship between 2 transcription factor, clock and BMAL1, and the cellular energy statue. This is crossing the observation of antibodies, breeding animal, for instance. I'm coming back to this data on the next slide, but you have different other food composition with which are important, like lipids, and in the field of research on preterm baby, you have a strong influence of the composition in protein. So next slide, please. Yes. Here we summarize the composition of the meal. So if you are breeding any kind of animal, you can synchronize the animal by distributing a low-calorie diet, because if the body is highly sensitive to the composition and the content of calorie in the diet, because it's a question of survival. After, in the lower level, you can have an influence with a high-fat diet or low-fat diet, and at in the third possibility, you have a low-protein diet, which can have an influence on the circadian rhythmicity, but this is on the acquisition of the circadian rhythmicity and limited to preterm and to babies as a general. Next slide, please. So this is a civilization. Next slide. In Europe, we have built up body mass index as a way to follow the health of population and to be able to spot people with obesity, for instance. And we have a definition which have been rediscussed recently by scientists in Hong Kong and US, because the body mass index, widely advertised in Europe over 30 as a signal of obesity for an individual, is not a world definition, because in China, for instance, they have described an explosion of diabetes with people with a body mass index below 30, meaning that you have people relatively lean and difficult to spot in a population in China. And the situation is similar in America with Hispanic American or African America. And the reason has been progressively described as a different composition in adiposity. So the adiposity is more visceral in some kind of phenotype in human population like China and America. And in Europe, we have a different way of defining, we have different people who are obese. And this is progressively shifting to a definition of the weight circumference. But of course, it's relatively difficult to measure accurately a waist circumference instead of a body mass index, which is relatively easy. Next slide, please. So I summarize on this slide the three kinds of diabetes. With the main type is type 2 diabetes because it concerns aging people, and it concerns also adults exposed to special type of work condition. The other type of diabetes, gestational diabetes, is known for woman and child, and the type 1 diabetes is special disease with genetic origin, and it can be related directly to failure of pancreas. But the most pernicious disease is type 2 diabetes, and this is what we are trying to fight in my laboratory and many others in the world. Next slide, please. Obesity is more or less linked to diabetes, and of course it's an excess of body fat. But the fat of the body is not always where you are looking for. So in Europe, we had a nice possibility to define an obese people by using his or her weight divided by the height of the person in meter at the power of 2. So you have normal body mass index and overweight or people too lean, below 18 of body mass index. The only problem is concerning obesity. This is not enough to define an obese on the world population as a whole. Next slide. So directly linked to the condition of work, you have sometimes metabolic syndrome. Metabolic syndrome is not a clinical state defined by a single disease like obesity or type 2 diabetes or heart failure. It's something mixed, and you have, of course, visceral obesity, but you have also dyslipidemia, hyperglycemia, hypertension, and this over the year, over 10 years, you are progressively drifting from a normal state to an individual developing diseases like type 2 diabetes, obesity, and heart disease. And this is especially relevant with premature baby. Preterm babies are frequently exposed to stressful situation because the mother is not always with them. We have no specific milk formula designed for the health of preterm baby. And so many people who have been preterm are developing a metabolic syndrome after 50 years. And the vicious side is that during 30 years, they are looking absolutely healthy and normal. But at around 50 years of age, all of a sudden, some small difference in their physiology related sometimes to the circadian rhythm, sometimes to other problems in their physiology, they are drifting to diabetes, obesity, heart disease, and this can reduce by 10 years their life expectancy. That means that if you are a night worker or if you are being a preterm babies, you have a higher risk to develop a metabolic syndrome than the normal population. And of course, you have a higher risk to have a shorter life. So a reduction of 10 years in your life expectancy. Next slide, please. So this is referring progressively to a concept of epigenetic instead of natural selection. So we are speaking of phenotype of people across a population, depending, of course, of their heredity and genotype, but the environment is of importance for them. Next slide. So this is how can an environmental event influence the phenotype of an individual, and is it possible that the influence on a phenotype before the reproduction of the individual can be passed to the children and grandchildren? So there is three main molecular possibility. You have the methylation of the DNA, modification of the chromatin on which the DNA is compact, and you have non-coding RNA, which are mediated methylation, but also directly interfering with gene promoter of methylation of histone, and they are disrupting the balance of your healthy phenotype and driving you sometimes to a disease. In the normal physiology, it has been shown that the fluctuation of some chromosomic region is related to epigenetic and to the circadian clock. And you can even link the balance of epigenetic with neural memory. Next slide, please. So on this slide, I am recalling you with the theory of natural selection from Darwin, and the possibility that this theory has limitation, and progressively we are in a situation where we have to recognize that the environment can influence heritability of some characters, and the information can have an impact on a generation, and this can be passed to the next generation through not mutation, but by epigenetic evolution. Next slide, please. So I'm presenting the way of a plant biologist, showing you on a small illustration that with the same initial genotype, you can select differently phenotype and future generation, future genotypes, according on the right to the Darwinian selection. You have a strict genetic point of view, and your phenotype is more or less the consequences of your genotype. On the left part of the dia, you can introduce the environment, and with environment signal, of course, you have a higher diversity of phenotype, and some phenotype can be passed to the next generation, and this is in a way showing some limitation is the theory of the natural selection, which remains true, but we can be better for the organization of our society if you introduce some epigenetic thinking in this way. Next slide, please. So here I'm showing you the life from birth to death, and it's like a spiral. And you have, of course, the mitotic epigenesis, but you have also during the development of an individual, being male or female, some specific point of time where you can have the environment influencing, programming the baby for his or her future life expectancy. But if the exposure to some environmental signal is during the perinatal window before the reproduction, it can be coded for the next generation and for the child and grandchild of this baby. Next slide, please. So here you are another illustration when you can see the ancestor, and I am working on breast milk and the importance of breast milk in the physiology of the baby, the capacity of this breast milk to influence the present day physiology of the baby, but also to program his physiology depending on the quality of the milk the baby is receiving. And if you are a term baby with a mother with a proper lactation, you are probably better fed than a preterm baby with a mother with difficulty to give you a lactation. And as we have no specific industrial formula to give to these preterm babies, the general health problem is that we have to realize more research and fundamental research to understand how is it possible to correct a phenotype which directly during the lactation period in order to obtain a healthy individual, even if the baby has been stressed during his or her life. And of course, it can have future consequences on the descendants of the baby, and progressively I shall introduce recent data from all over the world showing you this this kind of problem. Next slide, please. So the first demonstration, the most recent demonstration, has been published this year by ophthalmologists in the US, and they have shown for the first time, and I believe their work, that you can have a beneficial transmission from a stress during the life of a young mouse and before the reproduction. And if you are treating this adult mouse before the reproduction with some stress on the called retinal ischemia, which is sometimes applied to the human population, they have shown that the benefit of the treatment is not only for the mouse treated at the present day, but also for their babies and the future, the first generation of after the reproduction of these mice. Next slide, please. So an additional comment, it's on the slide, but anyway, so it the last time such a thing was shown was in 1880 with the work. Oh, sorry, I'm going to my English. Well, yes, it was some kind of calluses of the warthog during the Lamarck time. So the study of the ophthalmologist, if confirmed, of today, is a very important study in mammals, because it's beginning to show that we can, by manipulating the environment of an individual, not only to worsen his or her health for the future, but also to restore and to improve the health possibility of any mammals, hopefully with humans. So the rhythm of life and health has been changed since the invention of electricity. And of course, during the past century, we are in a society with a relatively easy access to food. We have also drastically shortened our sleep time, and for some time, for some profession, so for some professional like nurse in the hospital, you can be exposed to bright light during the night, and this can be a stress and with consequences on your health to develop a metabolic syndrome later. So we have finally to redefine the condition of life so that we can, for some professional of some ways of life, to change things or to have some cure available to be able to restore health or to preserve health. Next slide, please. My field is running under the hypothesis of the developmental origin of disease, and currently this hypothesis is telling you that epigenetic modification can retain the memory of the effect of environmental factor to which an individual is subjected throughout his life. And of course, because we are in the field of pediatrics, but this is a general rule, the crucial period is the early development. It can be prenatal at the fetal state, but it is also at the postnatal stage. It's especially interesting because when you have the baby, and not only the mother with the baby in the mother, it's easier to give directly a cure to the baby if there is any stress to prevent or to change. The epigenome is highly sensitive to the effect of the environment. And we can tell now that the individual are building a health capital during the 1,001st day of their life. And this is of importance not only for them, but also for their offspring. Next slide, please. So this is, I'm counting, so I have some time, still some time. This is to present you with one possibility to give to a small baby is to use microRNA. So microRNA are now considered a new micronutrient, and we have plenty of microRNA in breast milk of any mammals. And the fascinating things with breast milk is that you can formulate a breast milk based on the milk of cow, goats, and so on, and you can feed a human baby. And this looks natural, but of course, so we can replace partly the milk of the mother with the milk from other animals. But of course, you have regulatory molecules like the microRNA, which are conserved across the mammal groups, but with small differences and small consequences can have big impact on the health of an individual. So the microRNA are very small molecules. They are coding, non-coding gene in our genome. And in their mature form, they are around 20 nucleotides. And we have a functional side on the 5' side. This functional activity is widely preserved. But we have also on the other side of this molecule a zone of diversity, and this is a 3' side, and this diversity is used recently, has been used recently to demonstrate that some people drinking a glass of milk, of cow milk, are integrating in their plasma some microRNA coming from the cow with putatively physiological consequences. So the first demonstration has been made by Chinese scientists approximately 80 years ago, but we had a long history of discussion between scientists, and I guess that the paper I'm quoting of US scientist from Nebraska is demonstrating that indeed we can integrate some microRNA from milk and use with consequences on our physiology. Next slide, please. So this is to show you what a breast milk looks like in the laboratory. So it's on the right, it's a small tube with 1 milliliter. And we have approximately 900 to 1,000. It's always changing, never mind, microRNA within a breast milk. That means that we have 900 different molecular species. And of course, we absolutely don't know whether they are simply used in the catabolic way of nucleotide, only to obtain heat, or if some with a lot of microRNA concentration, with a high concentration of microRNA, or as the example I'm developing later, the microRNA 323P, which is moderately present in concentration, but which seems to have some impact on the physiology of the baby. And of course, the human cell, the somatic human cell, has much more microRNA, around 2,000, 2,500, and it's maybe not the end of the story, because the non-coding gene are really at the root of the phenotype of an individual. So we have, of course, a lot of information now on the coding part of the genome, but on the long non-coding RNA, the microRNA, the healthy RNA, we have a lot of questions to be solved to be able to understand properly physiology and to be able to prevent or to design new cure in the future. Next slide, please. So we should not forget that microRNA are very small molecules, and you have approximately 50 times more microRNA than a messenger RNA in a cell. So there is plenty of this molecule in a breast milk, for instance, and in other food, but they are not alone. They are very small, so the RNAs of the body, except in the digestive fluids, are not recognizing these microRNA as a target. And in addition, they are frequently complexed with other protein, argonaute protein, for instance, and they are in complex free in biological fluids like a drink, a glass of milk, and so on. So next slide. And they are going through sterilization. So this has been well shown by Chinese scientists some time ago. So that means that when you are drinking your milk, you don't need to be close to the cow. The industry is not, the industrial sterilization is not destroying such molecule. But of course, you have a minimal concentration to obtain a bio effect. And the minimal concentration has been set in vitro as a minimum of 100 microRNA of a specific form in a cell cytoplasm to yield a biological effect. If you are a lower amount, the message is not decoded and the organism, the cell, is not responding. Next slide, please. So this is only a small slide to recall you, but we apparently have a different behavior, different functional activity between plant and animals. But apparently, the more we are working, the more we are closing the gap between the plant, where the microRNA are supposed to degrade the messenger RNA, and the animals, including humans, of course, where we have only a subtle regulation of the messenger RNA, either inhibiting or stimulating the translation. So apparently the situation, this slide is relatively old, 2 years old, and we have probably in the next year, we have to show other data. Next slide, please. So if you are calling of microRNA. The best described activity of microRNA is cytoplasmic, because it's relatively easy to transfect or to deliver a microRNA in a cytoplasm. And we have a big discussion in the field whether the delivered exogenous microRNA is able to reach the chromatin, the DNA, so to cross the nucleus membrane or not. So on my experiment, apparently we have shown, like some other, that the delivered exogenous microRNA is crossing the nuclear membrane and can influence indeed the chromatin of the host. And you have not many up to now microRNA known for this activity. You have the 320 that I am discussing later, and some scientists in the US are exploring the chromatin-associated protein with the Rett syndrome, and the microRNA is 132. All of the microRNA are different activity. You have also the long non-coding activity, long non-coding RNA, which are involved in different syndrome. But apparently we have no, to my knowledge, there is no demonstration that you have a transfer from the cytoplasm of an exogenous long non-coding RNA interfering with the chromatin of the host and able to influence the acquisition of a phenotype. Next slide, please. The transmission of microRNA through breast milk has been suspected recently and reassessed by a Turkish scientist who are exploring the problem of babies from different litters, and of course with human, in many hospitals you have a lactarium, and it is of interest to know that in Muslim country they have a different point of view. They have affiliation not only of mother, father, and child, but also if you are sharing the breast milk of a mother, you are in a way becoming part of the family. And so that's scientists in Izmir who have designed a mouse model to obtain the possibility to show that when you are given the milk of a mother to baby coming from different litters, you are impacting the physiology of this small mouse. And the next slide is showing the main consequences. Next slide. I have a difficulty on my side, but maybe you can see the 24 months, which is the time of survival of the mouse. And they have found that you have a higher mortality in the group fed by a foster mother, by another mother than their own. And this is, of course, a data which should be confirmed because it has been recently described that you have small differences, but you should also consider that the health of in the mouse model has been impacted too. So apparently the group of baby receiving milk from another mother were in lower health state than the a group of baby receiving their own mother. So this is opening a new field and a new problem in facility where you are sharing the milk between different mothers, because as we are resorting to industrial formula from the cow, it is relatively little explored what the consequence on the long term of feeding a baby with an industrial formula, with the breast milk of his or her own mother, or on the mixture between an industrial formula and a replacement milk coming from a mother. So this is an open question, and anyway we have now a mouse model to tackle this problem. Next slide, please. So I'm going to be slightly more rapid. So I'm showing you now that we can either use a microRNA as a biomarker of physiology or as a new potential therapeutics. Next slide. So it has been shown a long time ago, but by instillating directly in the bloodstream of mice, that you have a functional effect. But if you are well, how can I say? It's far more easier to design a pill or a drink to be able to give orally to a baby or to any individual. And this is what we are trying to design. Next slide, please. So we have been helped by a lot of people working in diabetes and microRNA, and showing, next slide, please. This is only to illustrate the complexity. So people have explored of the blood parameter and the level of microRNA in the plasma of patients undergoing type 2 diabetes, type 1 diabetes, or gestational diabetes, and they have produced lists of microRNA in which you can pick the microRNA interesting for your specific problem. Next slide, please. In addition, in bariatry, in surgery, it has been shown that we have a wide gap of knowledge concerning the relationship between pancreas and the gut as a whole. And it has been shown that you can drastically reduce the problem of obesity by using a gastric shunt, a gastric shortcut in some patient with dangerous obesity. So it has been proposed as a cure, but it's showing indirectly that we can design some specific oral supplementation and target not only the intestinal cell, but also to change some function in the pancreas, for instance, of the host. Next slide, please. So next, this is a small drawing showing you on the left side all the possibility to engineer a vector way of transporting microRNA to the right side. So it's the body with a brain, with the blood, the lymph, and of course, we can easily introduce microRNA and vector by intravenous, but the problem of intravenous is that you need to be within an hospital, and when you have some kind of patient like preterm baby, they are already receiving plenty of intravenous injection. So it will be better to design either a topical, a cream, or some oral supplementation, so that it will be painless to introduce, painless for the baby, the patient, to introduce microRNA in the relevant cells. In my field, the relevant cells is the intestinal cell or the gastric cell. Next slide, please. So Sandra, yes, okay, next slide. I have to shorten next. This is to show you that you have, this is relatively important, different kind of binding site on the same messenger molecule, and you have a binding site on the promoter, on the 3' UTR, even in the coding region. And of course, depending on the organism you are using, you have, for the specific example of MIR320, a target very interesting, which is called R3D, which is the RNA polymerase 3, which is at the root of the regulation of many microRNA in our physiology. But you have also other molecules impacted if you are given MIR320 in a rat. And as we are working with a rat model, we had to tackle with a different binding site on the HSPB6. What that means, with small molecule, you can have many targets, and in the literature, you can read that you have hundreds of targets for a specific microRNA. In our modest experiments, we can say that you can screen the literature, and frequently the theoretical prediction are not so right. We have far less than 100 of target for a microRNA, but anyway, in the rat, for instance, we have to cope with 2 targets within the same rat. And of course, we are progressively designing experiment with the mouse, which is close to a human, and which is only where MIR320 is targeting only Pol R3D and not the other messenger RNA. So next slide. So only shortly, to show you that we have a specific vector, which is a natural vector derived from an antibiotic, paromomycin, and with chemists in Nantes, we can design a special type of vector so that we can engulf the microRNA and give the mixture to like an oral bolus to a small rat, and the rat is still bred fast, and so we are introducing the microRNA at a high concentration, so in the stomach, and we are following the consequences of this introduction on the physiology few hours after the introduction, and now up to 45 days of life of the rat. Next slide. This is to show that the consequences of this introduction is now a PhD of Gabriel Tavares in my lab, and we have been able to establish the list of the microRNA of interest with a target in the brain. with the dopaminergic and serotoninergic system in the brain of the rat, so that we can demonstrate that a stress like early weaning, if we are weaning a rat in half the time than in the natural situation, we are stressing the animal. And in Brazil, from where Gabriel Tavares is coming, they have experiments and data showing that you have the early weaning is a powerful stress on the brain balance for serotoninergic and dopaminergic. And so during the past year, we realized an experiment with Gabriel and many researchers in my lab, so that we can try to prevent the imbalance and to revert the phenotype created by this early weaning. And it happens for the time being. We have realized the experiments, and we have a communication in a congress last October showing that this bolus of specific microRNA can indeed have a benefic effect on the level of normalize the level of messenger RNA of the dopaminergic system for at 45 days of life of the rat after a single bolus given at 12-day-old under the mother. Next slide. And so we are close to the end. So this was to widen the discussion because a group of Swiss scientists is exploring the social links during the COVID outbreaks and the profile of microRNA. So it's an interesting review discussing all the different microRNA which can have some impact in the brain, and if the stress of social isolation can have or not an influence on the level of microRNA expressed in the model, mostly the model, because it's on rat and mouse. Next slide. So 1 hour close, I guess. So I can answer your question if there is any, or anyway, I hope that you'll be able to hear me comfortably, and that my French spelling was not too terrific for you. So, Sandra, back to your Spanish.
Thank you very much, Bertrand.
I can listen to Spanish questions if they are developed slowly.
Okay, exactamente. Gracias, Bertrand.
But I shall answer in English anyway.
Okay, well, don't worry.
Yes.
Es tiempo de las preguntas, justamente. ¿Alguien que quiera hacerle alguna pregunta al doctor Bertrand?
Can you repeat, because I had a problem with the sound.
Okay.
So can you repeat?
No, let me.
Okay.
Okay, well, let me. ¿Preguntas? Okay, well.
You have my email, so if you have some question, I can directly answer to the student. So Sandra can distribute my email. It's not secret. It's on the web anyway. And,
well, I have a question. En francés, bueno, en español.
As you like.
Okay. Well, thank you very much. For example, no, in the premature child, we can, well, I think no, this a little bit in research, no, to identify the markers for the healthy mother, and you can pass the milk at the premature babies.
We design a specific methodology in the hospital of Nantes to recover epithelial cells from the gastric border of the preterm baby. Because when you are a preterm baby, you're receiving breast milk with a pump every 3 hours. This is the same situation in Tijuana or in Nantes. So exactly the same in the hospital. So every 3 hours, a nurse is emptying the stomach of the baby. And so we design an easygoing system to isolate gastric cell from the baby. So in theory, but we are not receiving funding yet, but in theory, we can do manipulation on rat, and of course, in vitro, on the gastric cells grown from the baby, which frequently, you know, with so young baby, the gastric cells have some stem cell properties. And if you can tackle the problem of bacterial contamination, which are huge, you can grow cells from the baby and you can prove the rat system on human cells coming from a patient. But of course, we are probably a century before having the right to do it and its implementation and directly the baby. Well, but we have indirect methodology to prove that the system is working or not with gastric cell of a patient.
Okay, good.
It's working with an adult patient too.
Yeah, really?
You are equipped with a gastric tubes. You can recover, but it's with an adult is for problem of ulcer, and it will be interesting to deliver some drugs directly with to the ulcer. But with baby, it's only for the normal physiology of the baby to give approximately 20 milliliters of milk, breast milk or formula milk, industrial milk, depending on the health of the baby, every 3 hours. And so it's we have published curves of exfoliation years ago. And it's rhythmic because like in the 1960s, scientists have demonstrating that a baby has to be rhythmic. The circadian rhythm of the water loss of the skin should be organized. Otherwise, the baby is dying. And we have, in a way, shown that the gastric epithelium of the baby is rhythmic too. So in this publication of my young times, younger times.
Okay.
So maybe some other comment?
Yeah. Don't be shy. En España, bueno, en español, si quieren, le hacemos la traducción al. ¿Alguien? No.
I have no.
No.
I'm listening, but nothing is coming.
No,
Maybe
it's coming.
right.
Well, anyway, well, let me do.
So it's difficult by webinar. I prefer to be okay. Thanks. I prefer to be with real people because, you know, I'm talking with my hands. I don't know whether it's not very easy to talk with your hands in a webcam. So.
Yeah, I know. Well, from part of the Autonomous University of Baja California, we say thank you, Dr. Bertrand Kaeffer, for the talk, the diet and circadian rhythms. And I'm going to send you this small constance or certificate. And thank you so much.
So yeah, I have to thank Lise, Bianca, Adriana,